Okay, welcome to Antimicrobial Agents and Chemotherapy podcast. This is a new initiative from the journal, and this is the official podcast of the journal, and is going to be hosted by myself. I am editor-in-chief Cesar A. Arias from the University of Texas Health Science Center. And this podcast is intended to feature discussions with investigators and opinion leaders on the latest cutting-edge research and events related to the treatment of infectious diseases, including aspects of antimicrobial resistance, epidemiology of multidrug-resistant organisms, pharmacology of antimicrobial agents, and susceptibility testing, among others. As we are now currently in a big crisis in epidemic pandemic, our episode 1, and this is going to be our first episode, so we are also making history with AAC, and it's going to be entitled Therapeutic Approaches for COVID-19, Myths and Facts. And in order to discuss this, I have two very distinguished investigators and academicians and doctors that are going to tell us a little bit about what is happening in this field of therapeutics. So it's my pleasure to introduce Dr. Kalil, who is professor of the University of Nebraska Medical Center, Omaha, and is the lead investigator of the NIH clinical trial for COVID-19. I'm also very pleased to introduce Dr. Miguel Martinez, who is an editor of Antimicrobial Agents and Chemotherapy, and he is our virology section editor, and he is at the AIDS Research Institute-IrsiCaixa and Hospital Universitari Germans Trias Pujol in Barcelona, Spain. Welcome, gentlemen. Thank you for your time.
Okay, thank you, Cesar.
Thank you.
Nice to meet you. Welcome.
So the idea in this podcast is to, try to discuss a lot of the data that are coming, either supported or not, on treatment of COVID-19. So the first question I want to Dr. Martinez to introduce the topic. So from the virology point of view, what are the bases of therapeutics against SARS-CoV-2? And basically, how do you target this in the virus and the anti-inflammatory response?
Well, first, this infection, we have two different periods. We have the first week of infection that we have a virus growth and clearly, let's say, a virologic part of the infection. And after 1 week, more or less, of the infections, there is an inflammatory response. This inflammatory response in 5 to 10% of the infected individuals is pretty strong, and is the reason why this percentage of infected patients develop an important inflammatory disease and causing a high morbidity and mortality in this percentage of patients. So clearly, the first aim, to try to stop this virus infection, is to have a specific and a strong antivirals against the virus. To avoid this second phase of the infection, this inflammatory response of the infected individuals. So, in my opinion, the aim of the virological research in this area is to try to stop the viral replication at the beginning of the infection. And well, we can discuss later on that still we don't have a good specific virus individuals, to stop this phase of the infection. And so now in this pandemic, there are many anti-inflammatory drugs that are being used in this, let's say, second phase of the infection. But these anti-inflammatory drugs are not directed against the virus, but are rather palliative, drugs to try to stop this high inflammatory response.
So, what part of the virus replication machinery is being targeted now with the antivirals that are now in clinical trials?
Well, well, in my opinion, well, there are now a few antivirals that are not so specific. So what we are doing is to try to repurpose antivirals directed against other viruses to try to see if they are effective against this virus. So we are trying to use protease inhibitors of the HIV protease. We are trying to use nukes, nucleoside analogs targeting other RNA viruses like Ebola. And although still we have to wait for the results of several trials running now, in my opinion, they are not going to be so effective, and they are not going to be so specific. I mean, to stop viral replication. What I mean is that it's not clear that those antivirals running now in trials are specifically targeting the virus.
Okay, very good. Dr. Kalil, you've had experience with this sort of epidemics and therapeutics before. And of course, there are many compounds there that are being used. And the data behind using these compounds are pretty weak at the moment. And you recently had an interview in the New York Times where you mentioned these emotional issues of your compounds. So what is your opinion of using compounds that are not proven to work in epidemics, bearing in mind that many, for example, hospitals do not have access to clinical trials?
Thanks for the question, Cesar. It's always a challenging situation, being in the middle of an outbreak and not having any specific therapy, specifically because we're dealing with the infection that we never dealt before. It's a new virus, and it's everything pretty much having to be start from scratch. It's we have the advantage through the years to find new therapies for bacterial infections, viral infections, parasitic infections, fungal infections, but we have the leisure of the time to do the clinical trials. While in the middle of an outbreak, we really time is against us. We have to be way faster than ever before, and I think this is definitely the biggest challenge. Now, that being said, I think this is something. Your question is very, very important for the moment, because if you don't have a trial in a place that you are practicing now, your biggest job is to not harm your patients, and that's the way that I treat all my patients. First, do no harm. And what I mean about first do no harm is that we cannot simply, in the name of the really difficult situation, start giving drugs that not only are unproven to these patients to treating this type of infection, but most of these drugs, even drugs that we've used before, they have absolutely no data on safety. And why this is important is because safety of a drug in a context of one infection does not translate into the same safety in the context of another infection. I'll give an example. If you give, let's say, if you're trying to repurpose a drug that was used, for instance, for patients with malaria, like chloroquine, or patients that have rheumatoid arthritis with hydroxychloroquine. This patient population, either with a chronic rheumatoid arthritis or with a malaria, is very different from the patient population that actually is getting sick with COVID-19, the ones that have the highest risk of complications. Why I say that? I say that because that's even though it sounds obvious, but that's very, very important. Patients that have received malaria for the last 50, 60 years with these drugs, they are usually young patients without comorbidities traveling around the world. Patients that receive drugs for rheumatoid arthritis are chronic patients, stable without any acute illness, with joint inflammation and with inflammation that can be subdued with the medications that they take, like hydroxy or some of these IL-6 inhibitors. So these are not the same patients that are getting sick with pneumonia, with an acute viral pneumonia, critically ill, and requiring a impressive amount of supportive care. So these patients have not only they are older with COVID, they are older, they have more chronic medical problems that include chronic heart problems, chronic liver problems, chronic kidney problems. And on top of that, they are mostly in a hospital or in the intensive care unit, receiving acute care because of the critical illness. So you have a patient that's older than patients with malaria, that a patient has more chronic comorbidities than most of the other patients, including rheumatoid arthritis, and patients that actually are critically ill with a acute pneumonia. So translating any drugs from the past to this kind of situation, it's dangerous because these drugs were never tested in a critically ill infection like COVID-19. Let alone COVID-19, but not even in a critical ill patient. These drugs have never been treated, never been given in patients with a critical illness secondary to infection, let alone COVID-19. It's a brand new infection. Why I'm saying that? I'm saying that because these drugs have severe side effects, and these side effects actually can increase the mortality on top of the mortality caused by COVID-19. So the moment we start to give any of these off-label drugs, or experimental drugs without having a control, you run a risk of actually harming your patients. And on top of that, the outbreak is going to end, and we're going to learn absolutely nothing about what can treat this infection. So a few months from now, this coronavirus comes back in the winter time, or comes back in a second wave, or maybe in another strain with some mutations. And then we look back and say, we found nothing, we have no treatment, we're going to start again giving the same drugs. So if you want to really provide safety to your patient, provide supportive care that's necessary, that's the only thing proven to save the lives of patients with severe COVID-19, or try to enroll your patients in a clinical trial. But I would really recommend against any kind of off-label, any kind of drugs that don't have safety, because you really want to make sure that you do not harm your patients.
I will get back to that point, and thank you very much. So, Dr. Martinez, you mentioned about these viruses or these antivirals being used for other drugs trying to be repurposed for SARS coronavirus 2. So, can you explain the difference, for example, in therapies that were used for SARS with SARS coronavirus 1, and why those will not be as effective for SARS coronavirus 2? What are the main differences between those, and why do you think they will not be as effective?
Well, in terms of the main targets of the virus, SARS-1 and SARS-2, I'm thinking about the RNA polymerase or the main protease that clearly are probably the best two targets of these viruses. We don't have any drug against SARS-1. We have some data from in vitro studies targeting SARS-1, or we have some data and some animal models that show some efficacy of some drugs against this SARS-1. But unfortunately, we didn't have any clear, potent and specific inhibitor of SARS-1 virus. So that's the reason most of the current trials are going to be disappointing, because we don't from the very beginning we didn't have any potent SARS-1 inhibitor, and without any clear clinical trial showing the potency of this hypothetical inhibitor. That's the main problem. In terms of RNA polymerase, protease, both viruses, they have a very similar proteins. So probably if we are trying to target the binding of the receptor of the virus, probably there is some differences between SARS-1 and SARS-2. but the problem is that we didn't have any specific good inhibitor for SARS-1. That's the problem.
Okay, very good. Dr. Kalil, you are leading one of the largest clinical trials funded by the NIH. Can you tell us about the trial design and what drugs potentially are going to be studied? And I imagine how the recruitment is going, whatever you can share with us. And when do you think we will have some potential data that could inform us about therapeutic approaches?
Sure, Cesar. So the trial is a randomized, adaptive randomized, double-blind, placebo-controlled trial. It's a loaded title, but it is what it is. So it's the randomization, meaning that half of the patients are planned to receive remdesivir and half are planned to receive the placebo. That's identical infusion, 30 minutes a day for up to 10 days. It is adaptive in the sense that remdesivir is the first drug that we chose to be part of the trial, but the trial is open to any therapeutics while the trial is up and running. And I'm gonna give a little more details on this, but the goal here is to really develop as a platform in which we can test several drugs either sequentially or at the same time. It is double-blind in the sense that both the healthcare providers and the patients do not have access to know what's being infused, either the experimental drug or the placebo, and this is really, really important, especially in a situation like today during the outbreak, because once healthcare providers know what's being given to the patients, this can have a substantial confounding effect and actually can undermine the reliability of the study. So that's why it is very important that we have the randomized double-blind placebo-controlled design. The study was planned to enroll approximately 500 to 600 patients in order to have around between 400 and 500 patients available in terms of attrition rate, in terms of missing data. And the first part of the study, the remdesivir part, is already concluded. We concluded last week, and we enrolled way above actually the sample size that was predicted, just because the enrollment was way faster than we ever imagined. So at this point, we are finishing up data collection and starting data analysis. And the plan is that in between this week and next, we are looking into the preliminary analysis, and the total final analysis of all patients should be coming out between mid-May and between mid and late May. So in the next 3 to 4 weeks, we should have a lot of information coming out from this trial. And so in the trial actually is the first part is completed, and the goal now is actually, while the trial is being evaluated, is to add a second therapeutics. And I think Cesar, you have a plan to discuss a little more about that. It's baricitinib. It's a anti-JAK inhibitor that's going to be added with and without the remdesivir, but we can talk a little more about that. But this is going to be the upcoming second part of the trial while we are doing the final analysis of the remdesivir versus placebo data.
Okay, well, that's very hopeful. That it seems like we will have some data very soon. I think I want to continue with you in terms of the combination therapy. As Dr. Martinez mentioned, this biphasic phase or the pathophysiology phase that involves the antiviral targeting and possibly inflammatory targeting is one possible alternative. Of course, in the second is a very complex issue because there are many pathways of inflammation. Many of these anti-inflammatory drugs have been tried with, I would say, very weak data that actually are going to work. So how is going to be decided which drug or which particular pathway is going to be move forward in the trial after the first step?
So Cesar, we looked into a multitude of medications. As you know, there are a a numerous number of anti-inflammatory molecules. Most of them are being used for patients with rheumatoid arthritis, and so as you alluded, it's very important what you brought up, Cesar, is that some of these anti-inflammatory interventions have been used in the past for patients with sepsis, especially bacterial sepsis, and without too much success. And actually, and some of these old molecules actually potentially provided some harm to patients with sepsis. The idea is that sepsis was pretty much a cytokine storm type of disease, and this is what now is one of the assumptions, that some of these patients potentially could be dying from a hyperinflammatory response to a cytokine storm. So we gotta be very cautious about this. I think that the history on infectious disease and immunomodulation is not very happy. We've never really found immunomodulatory drug that has proven to change beneficially the outcomes of infectious disease in acutely ill patients. So this is very humbling time, very difficult, challenging, but there's no questions that there are patients that are having this hyperinflammatory response. We don't know what's the proportion of patients that actually are having this response, and then this is another challenge. We don't have so much of the natural history that we'd like to have in patients with COVID-19. But that being said, this is something very important, is that this has to be done very carefully, because if you over immune suppress, because the moment that you bring the inflammation down, you will bring the immunological response down as well. So you cannot separate both, right? So inflammation is part of how we defend ourselves against infection. So we cannot be that simplistic, say, well, you bring the inflammation down and life is good. Well, you bring the inflammation down. If the inflammation down means immunological response down, you may end up harming, because immune suppression is not what you want in somebody that is critically ill with a viral infection. So we have to find which patient population potentially can benefit from this, and which medications will quiet down that inflammatory response in a way that is not going to over immune suppress and cause harm to the patients. And that's why I do not recommend any of these drugs outside a clinical trial, because we already know these drugs. If you read the label of each one of the drugs, the approved label, they all can provoke significant immune suppression. So if these drugs are to be evaluated in acute patients with COVID-19 pneumonia, the only safe way to evaluate these drugs is within the context of a randomized controlled trial, because we're going to have the comparison, we're going to have the safety monitoring. If there's something that does not, there's any signal of safety, the trial is going to be able to detect and stop. If there's signs of benefit, even better. But giving these drugs outside a randomized trial, knowing these drugs can be quite immune suppressive, to me brings serious risk of safety to our patients. And that's why I think that if we're going to move forward with this, as we are moving forward, both in our trials and other trials, this has to be done only in the context of a randomized clinical trial.
Excellent. Let's switch a little bit gears and talk about vaccines, because, of course, the hope of controlling this eventually is to have an effective vaccines. And there are many efforts around the world to try to develop a vaccine on this. Dr. Martinez, can you tell us the broad sort of strategies that have been put forward to develop a vaccine, and what are your thoughts about when we can have a vaccine, and what will be the challenges that we will face when the vaccine is available to administer in grand amount?
Well, first, it's important to say that we have a new virus. So there are, and that is a very important issue, because we cannot anticipate many things. So now there are different kinds of vaccines that are being developed, and in principle, many of them can be pretty safe, and even they can be quite good vaccines. the only thing that we don't know is exactly what is going on with this virus infection. So we don't know the memory. We don't know if the vaccine memory is going to last. I mean, to protect for months, years, or weeks vaccinated individuals. So this is, in my opinion, my first opinion. Also, we have to be sure that the vaccines are going to be safe, in terms that we don't know very much what happened with reinfection of this virus. So probably in the next months we are going to know individuals reinfected with this virus. So we are going to know if the reinfection is being good or bad for that individuals. I'm thinking in some viruses like dengue or other respiratory virus that, second infection sometimes or memory against the virus can be dangerous. So first thing, like just told us Dr. Kalil, I agree that safety is one of the main issues. And well, if things run properly, we can have in 1 year or 1 year and a half, we can have a vaccine of different kinds. There are subunits, inactivated vaccines, mRNA vaccines. Most of them, they can be pretty okay. I don't see this virus mutating like other arenaviruses. I think in flu or other HIV or other or ACV virus. And I think that this virus is more conserved. Probably this is not going in principle to be a big issue, but first we have to know better the infections, and we have to know which memory we have and which is the memory that we need to protect individuals.
So just a short comment about the inflammatory response and the vaccine, Miguel, taking the example, as you mentioned, of dengue, that possibility that there may be differential efficacy in people who have had the disease versus those who did not have the disease. Do you think something very similar may happen with a vaccine for SARS-CoV-2?
Well, probably not, but we have to be sure that is not going to happen. So we have there are animal models, and and we are going to know that in the first clinical trials. But we have to know that because this is a pandemic, we are going to find individuals infected twice with the virus. So we have to study these individuals and to know if there is a toxic effect in this memory against the virus. But in principle, it's a safety issue that we have to take in account.
Yeah. A final question for Dr. Kalil. What do you think about drugs for prophylaxis? And do you think there is? I know there are clinical trials trying to address this. And what is your opinion on the clinical trials and the design of these studies for prophylaxis? Something like, sort of PrEP that is very common now in HIV and shown to work.
Thanks for the question, Cesar. I would love to have a prophylactic regimen for for this disease, especially in a situation where all of us are taking care of this patient on a daily basis, healthcare providers, first responders, police, EMTs, so many people that are in the front lines that could benefit from a prophylactic regimen. I think that's absolutely very, very important question. At this point, at least, we don't have any drug that really has proven to have efficacy against COVID-19 in humans. I mean, as Miguel mentioned, as we talked before, we have in vitro data, We have some animal data, but we really don't have. So I think that the difficulty that we're facing here is that if we don't have anything that actually is known to treat the virus, it's just hard to really pinpoint and find drugs that can be used for prophylaxis. Because remember, what happened with prophylaxis is that you have to have a very good safety profile, because you're giving drugs to healthy people that do not have the disease. So your risk-benefit profile here has to be different. And somebody that's critically ill, the risk-benefit profile is very different from somebody that's healthy that has no disease. So you want to make sure that safety really becomes key here. So there are several trials being done with different medications, but at this point we don't have no results. So we have no results, at least from any of these prophylaxis trials. But I want to mention, and that just a segue to what Miguel just talked about. I think that there will be absolutely nothing better than a vaccine to really be the best prophylaxis for these patients. That being said, I wouldn't be surprised that we may end up with something more novelty in the next year or two. There are several companies working on polyclonal and monoclonal antibodies, in which they not only they potentially have the activity, against. They're going to have to be tested, sure, in vivo and treating patients with COVID-19, but several of these formulations, they can last long enough that potentially can be useful as prophylaxis, especially for the first providers and first responders and healthcare providers. So we may end up with a combination strategy between vaccines and antibodies, that potentially can both have different effects in the long term for patients with COVID-19 on top of medications. But at this point today, we don't have any medication that is safe and efficacious to prevent the acquisition of COVID-19.
Well, I want to thank Dr. Martinez and Dr. Kalil with this wonderful conversation. Lots of things to talk about, and I think this is very informative, and I appreciate your time. Thank you for our first broadcast and ASC podcast, and I want to invite you for the episode 2, where we will discuss some about diagnostic challenges for COVID-19. This is Cesar A, editor-in-chief of ASC. Thank you very much.
Thank you, Cesar.